Okay, so that brings us to genes. So each chromosome is made up of genes. We talked about that earlier. You got lots of genes on each chromosome. So you think of like a necklace, and each individual bead is one gene. So it takes a whole bunch of beads to make up the one necklace, the one chromosome. So the gene for each of your traits, like tall or short, or blue or brown, or whatever, is going to be on the exact same spot on this particular chromosome. That's called a locus, the gene locus. Okay, so when they mapped, we talked about the human genome in the last section, and the human genome allowed us to know where each gene is located and on which chromosome. So the human genome actually helped us learn the locuses for each particular gene, human gene. So anyway, genes for the same trait that have the same locus, but it might be on, different homologous chromosomes. Those are called alleles. So if it's the same trait, same locus, but remember you have your paired chromosomes, so you have two number 22s. One came from your mom, one came from your dad. Those are going to be called the alleles. Okay, so getting into Mendel's theories and laws on heredity. Some of these are laws, or they're called laws because we can prove them. So anyway, the first one we're going to talk about is the law of dominance. So we've already mentioned the law of dominance. That's basically saying that you have dominant genes and recessive genes, and the dominant will hide the recessive. And so that's if you have two contrasting or alternative traits. So anyway, one of the other ones is that the first law is very simple. It's that heredity traits are transmitted by genes which exist in pairs. Y'all already pretty much understand that, but it's just saying that all your genes are paired up. So you have two letters for each particular gene, and you got one from each parent. Now, the law of segregation specifically is going to talk about chromosomes during meiosis. This is very easily confused with the law of independent assortment, because in both laws they're talking about things separating independently. So to segregate means to separate. So if you think about back in the time of segregation, that was when black people and white people were separate. They had separate schools and separate water fountains and everything. And so that's kind of the most common usage of the word segregation. But in your body during meiosis, your chromosomes actually separate. Okay? So when gametes are formed, that's meiosis. Your alleles for each gene, okay, so those little letters, the big T, the little t, are going to separate independently of one another. Okay, so the big T is going to go in some of the eggs, and the little t is going to go in some of the eggs if you have those two letters. So basically, your sex cells are only going to carry one letter for each sex cell. Okay, so if you take a look here, remember in interphase, in the S phase of interphase, what happens in the S phase of interphase? Hopefully you remember that DNA doubles, right? It replicates. Okay, so my 46 chromosomes become 92, right? So then in meiosis 1, it gets split into two cells with 46. So we split off the alleles. And then in meiosis 2, at the end of meiosis 2, you have 4 haploid cells. Okay, so they will only have one letter each, one chromosome each. Okay, so you'll only have one letter, even though normally you would have two. So this person's genotype would be big G, little g. They doubled it here, in the S phase of interphase, and then they eventually split out into 4 haploid sex cells with 1 letter each. So this is just showing you that if dad has genes A and B, mom has L and M, the baby might have, different combinations of that. A from dad, and then M would have came from mom. So then that brings us to the law of independent assortment. So it's saying that the genes of different traits are going to separate independently of one another during meiosis, during gamete formation again. So you can see how the law of independent assortment and the law of segregation are very, very closely worded because it's both talking about separating independently. Okay, but what is different here is saying that like blonde hair gene, so genes of different traits, the blonde hair gene is going to separate separately or independently from the blue-eyed gene. You've ever heard blonde hair, blue eyes? So that is what the law of independent assortment says. It's saying that all your different traits are going to be separated out differently or independently during meiosis. So both of this is going to happen during meiosis, both of these laws. So technically speaking, inheriting one particular trait is not going to be linked to the inheritance of another trait. Now, this is what Mendel talked about, okay? So this was what Mendel found when he was studying pea plants. So he said that the alleles for plant height were not correlated in any way to the flower color. So like brown eyes doesn't have anything to do with freckles, or blonde hair and blue eyes, there's not a connection. But there have been some geneticists that have come along after Mendel and done some research and found this is not exactly the case. So we know this applies to only to genes that are on different chromosomes, or that are further apart on the same chromosome. If it is on the same chromosome, then there is such thing as genes that can be inherited together. Okay, so this is just showing how you can, if you both parents are hybrids or heterozygous, we could have pretty much any different combination in the offspring. Okay, and then this is showing how in the egg and the sperm, you will only have a haploid, so you will have half of the normal number. So you'll only get one allele. So then the baby, so this would be like a haploid plus your haploid restores your diploid in the baby, the 2 letters. Okay, and this is just another picture again showing that, this letter could go into any half of the offspring, and this letter could go in the other half. And so you could see babies that, if you have 2 heterozygous, then you could have all 3 possibilities: big A, big A, big A, little a, or little a, little a. So I mentioned a few seconds ago that there is a such thing as linked genes. So like I said, Mendel, he was one of the earliest people who studied genetics. There's been people, of course, that have come behind him. So Thomas Morgan, and again, anytime you see someone's name, you need to know, what they did. But Thomas Morgan, he also had students, graduate students that worked with him, but they came across linked genes. So Drosophila, you see that word a lot. Drosophila is a fruit fly. And fruit flies are very commonly studied because their life cycle is extremely short. They usually have like a 24-hour life cycle. So the common fruit fly is extremely popular to study by geneticists, and that's called the Drosophila, and that's why you see Drosophila so much in biology. So Thomas Morgan, his graduate students, they were studying the Drosophila, and they realized linked genes are a thing. And so what that is if you have your chromosome here, so if this is my chromosome, and crossing over is going to happen. So when crossing over happens, it might split equally, and move into, hang on, let me do this a little better. So if crossing over happens, it might split, and you just get half and half, or this part might swap, right? So if the yellow, if you swap this yellow part, and there's a gene here and a gene here, they're very likely to be to come together, to be inherited at the same time, inherited together, because they're so close together. So remember, you have one chromosome. So if you think of a chromosome as like the human body, and you made an X shape with your body, your genes on your hand, okay, you got 5 fingers on your hand. Let's say there's 5 genes on your hand. If the whole hand goes, when crossing over happen, if the whole hand gets swapped with the other hand on the other chromosome, then those 5 fingers, those 5 genes are going to go together. They're going to be inherited together. So if, one is blonde hair and one is blue eyes, then those do tend to be inherited together. So, that is what Thomas Morgan discovered, was linked genes. So these are genes that are located very close together on the same chromosome, and they can then be inherited together. And this is just showing a fruit fly. The Drosophila, which is extremely common to study in genetics. Blonde hair, blue eyes do not correlate for humans. One of the reasons for that is because there's more than one gene that controls both hair color and eye color. Okay, Gregor Mendel pioneered studies of heredity in the 1800s. He believed that parent organisms could transmit any random combination of characteristics to their offspring. Today, however, scientists know that some of the parents' characteristics are inherited together as a group because A, certain genes attract one another and then stay together. B, many genes are located together on the same chromosome. C, pairs of chromosomes are joined together like 2 sides of a zipper. Or D, the chromosomes from one parent are dominant and those from the other are recessive. Okay, so this is saying some of the parent characteristics are inherited together. Okay, so what does this make you think of? Hopefully it makes you think of linked genes. And so B would be the correct answer. Many genes are located together on the same chromosome. So that brings us to Punnett squares. So a Punnett square is just a diagram that predicts the possible babies of a cross, so the offspring of a cross. So I can do a Punnett square between 2 people and look at the possibilities of those 2 people's children. So the simplest Punnett square is 4 boxes inside of a square, and that is a monohybrid cross. Okay, so what you're going to do is on one side, on the top, and then on the other side on the left, you'll put the possible gametes for each parent. So you can put the female on one side, the male on the other side. And so the possible gametes, those are going to be like what is going to be in the sperm and egg. And if you recall, those are haploid. So anyway, each box inside of that square is then filled in by bringing the letters down and over. And so all these letters you use in the Punnett square, the boxes on the inside, but also on the outside, those are the genotypes. Okay, so the inside is the genotypes of the children, the possibilities, and then the outside is the genotypes of the parents. So here's a couple of examples of Punnett squares. So if you look up top here, I like this one because it shows you red and blue. Blue for the dad and red for the mom. So it shows you how we have this could be a possibility for sperm. This could be a possibility for sperm. This could be a possibility for an egg. This could be a possibility for an egg. And then it shows you how this A is brought over and over. This one is brought over and over. This one is brought down and down. And then if you look over here at this Punnett square, they did the same thing, color coding red and blue for red for the mom and blue for the dad. And you can see that this big D here is brought down, and this little d is brought down, and this is brought over. So this letter comes down, this letter comes down, this one comes over, over. Okay, same thing here. They just didn't color code it. And then, of course, the inside, this would be the offspring. These would be parent 1, parent 2. And then this is a dihybrid cross, which we're going to get into. You just wait. So the dihybrid is looking at more than one trait. So if you look here, they could either be round and yellow or wrinkled and that says green. So it's looking how 2 traits are inherited rather than just one. Okay, so again, a monohybrid cross is a cross involving one mono for one pair of contrasting traits. Okay, so for example, in this case, they're talking about tall and short. So the contrasting trait is height. Okay, so it's either tall or short. Those are contrasting. Okay, so we went over some terms earlier about homozygous and heterozygous. And just a refresher, hetero is different. So heterozygous is going to be usually a big letter and a little letter, 2 different letters. So here will be our heterozygous. So it says cross, our example is to cross heterozygous tall. So the first thing I'm going to do is I'm going to make myself a key. These are my possibilities for T because it tells me that I'm going to use T's here. Doesn't always tell you. Sometimes you'll have to figure out the letter on your own or just make a letter on your own. It doesn't matter because the question might just be asking, you know, what the offspring are going to look like. But anyway, it tells me T is dominant for tall. So it tells me big T equals tall and little t equals short. So I know these 2 will be tall because they have a big T. And the only option is short because it's 2 little t's. So it says cross heterozygous tall. So heterozygous, like I said, is a big and a little. And remember that big T codes for tall and the little t codes for short. But the big T is dominant over the little t, so this organism will be tall. So anyway, heterozygous tall. So homo means same, so it's going to be the same letter. And it's going to be short. Well, our only option for short is little t, and that is homozygous. Okay, so we're going to take one parent, put it over here, one side, and then the other, put it across the top. And then I bring it this letter over, this letter over, this letter down, and down. Okay, and I'm going to erase all that just because it's looking kind of busy. Okay, so on the outside, again, we have our parents. That's what's in the gametes. And then the inside is going to be the offspring. Okay, so these are your possible offsprings. So these 2 will both be tall. These 2 will be short. So I have my genotypes. So a lot of the times you'll be asked to give this information here. Okay, so looking at the inside of the Punnett square, the possible offspring, I have 2 that are big T, little t, and 2 that are little t. Those are my genotypes because they're the alleles, they're the letters. Okay, my phenotypes is the physical appearance, so that is tall and short, and 2 of them will be tall and 2 of them will be short. So this is, you might see it asked as a ratio. So you would put how many of each, and pretty much you have a colon between it. It's kind of like a fraction. But a 2-to-2 ratio can be reduced to 1-to-1. So, of course, your genotypes will be 2-to-2 and your phenotypes will be 2-to-2. And then tall and short. Well, 2 out of your 4 are tall, so that's 50%, and 2 out of your 4 are short, so that's 50%, because there's 4 total boxes up here. So that would be basically how you do a Punnett square. So looking at this one, if you would cross a heterozygous tall plant with heterozygous tall. So if you're crossing heterozygous and heterozygous, you're going to have the same genotypes. So you're crossing 2 with the same genotype. Okay, so both sides will look the same, but the offspring will look different. Okay, so this one, and this one will all be tall, and this one will be short. Because remember, we already did a key, and these were tall, and this was short. So in this case, you have 3 genotypes. So if you're looking down here, you have 3. 1 that is big T, big T, 2 that is big T, little t, and 1 that's little t, little t. So that makes your genotypic ratio 1-to-2-to-1. Now, you only have 2 phenotypes, tall or short. So you'll always only have 2 numbers in the ratio. And we already said that 3 will be tall, 1 will be short. So 3 out of 4, 1 out of 4, 75, 25%. And again, you're just bringing those numbers down for the ratios. So your genotypic ratio, sorry, I accidentally changed the slide. So your genotypic ratio is going to be the number 4, big T, colon, big T, colon, little t. That would be your genotypic ratio. So in this case, it'd be 1-to-2-to-1. Now, if we don't have, let's pretend we don't have any of these. So I'm just going to cross that out. Well, then it still has to be 4. The ratio's got to have at least 4. So maybe it's 3-to-1, which doesn't happen, but let's just say 2-to-2. I just wouldn't have to put anything there for that other one. And in phenotype, so I have 3 tall to 1 short. So your ratio would be tall, colon, short. The number of tall, colon, the number of short. Okay, next example, homotall. Again, I'm going to write our key up here because that takes just seconds to do, and it's very beneficial. So if you're homo for tall and homo for short, then you're going to have one that has 2 of the same big letters and one that has 2 of the same little letters. So put them on the outside of the Punnett square. One parent here, one parent here. Doesn't matter which side you put it on, top or left. It is usually on the left, not on the right. But anyway, when we fill these in, we bring this one over and this one down, and the big T's will always come first. Then you will get your answer here. And all of the offspring are the same. They're all heterozygous. 4 big T, little t's, and 4 that are all tall. And so you would write the ratio 4-to-0 because you need at least 2 numbers in a ratio. And of course that would be 100% tall, 0% short. Anytime you cross a homozygous dominant with a homozygous recessive, you are going to see this ratio. Okay, and another one. Again, going to put our key up here. Which takes just seconds. So homotall, so homozygous, same letters, and they're tall, so that would be big T, big T. And then hetero, my only option for hetero is big T, little t. Okay, so it might help you to even label it like this. Homozygous dominant, homozygous recessive, and heterozygous. Some people, when they make their key, they add those words in. It's just up to you. So anyway, put them on the left, one parent on the top, one parent on the left, bring it down and over. This is what you get. You get 2 that are homozygous dominant and 2 that are heterozygous. Okay, so here are my genotypes. 2 big T, 2 big T, little t's. So all of those will be tall. All 4 will be tall. So again, you'll notice the ratios are a little different because my genotypes are 2 and 2, but my phenotypes will all be tall, 4 to 0. Okay, and this one is a little bit different. Okay, so you have a key here. They've already given you the key. Now, this particular trait is actually considered non-Mendelian, okay, which means it doesn't go by typical dominant recessive rules, the law of dominance. So what you have here is a third phenotype, okay, wavy hair. Okay, so when it's a mix like that, it's called incomplete dominance, which will be we'll be going over that later. But anyway, so if it's hetero, then that's going to have heterozygous is going to have a big letter and a little letter. Might be healthy. And then homozygous recessive, that would be your 2 little letters. Okay, and so we'll just put that there. So anyway, when we cross these, you still are going to get 2 that are big S lidless and 2 that are lidless lidless. And so if I look back at my key, I can see 2 have curly hair and 2 have straight hair. So we have it. So that will be a 2 to 2 ratio, which remember you can reduce to 1 to 1.